Good evening, dear colleagues. We are starting our webinar of Ovogene Egg Bank. This is our first webinar in 2021, and I would like to welcome you in this new year and wish you all year to be full of joy, health, happiness, and all great achievements which you wish to happen. I would like to ask you to unmute yourselves in order for us to start, so it will be great to listen the speakers. My name is Dr. Uliana. I'm the medical director at Ovogene Egg Bank, and my colleague Birol Audin, the clinical laboratory manager, embryological supervisor, and Ovogene Egg Bank scientific advisor, have prepared for you presentations, and the topic is donor recruitment, legal aspects, algorithm inclusion and exclusion criteria. Please let me know if you may see my screen. I hope it works. Yeah, so we are ready to start. Before we start talking about the oocyte donors, I would like to start talking about... I'm sorry, Victor, can you please unmute yourself? So we have the total number of couples around the world experiencing fertility problem is 90 million people. And even if we will perform a simple calculation that 1/3 of those will search for the fertility solution for the treatments, and 50% of them will decide to be seen by the IVF specialist and dedicated for that, and 10% of those will need an oocyte donation treatment as a solution for their fertility problems, we are ending up with 1.5 million people who need oocyte donation annually. And as we are talking now from Ukraine, and Ukraine is not a European country, but we are very close located to European Union, and we are included into their registry. I would like to specify from the registry, based on the figures, that in the past 10 years, the demand in egg donation have tripled. And this is just up until 2015, and until now, probably it increased even more. Ukraine, as you can see on that map, is always in the map of providing oocyte donation, also for Ukrainian patients, and also for foreigners. Of course, we are not as huge as Spain, and the main treatment options were provided as patients visited Ukraine, not for local patients, but for international. And in this data from ESHRE register, you can see the total number in all countries where oocyte donation was performed. This is a bit more than 60,000 egg donation treatments. And Ukraine is mentioned as providing over 1,000 treatment cycles. However, we know for sure that in Ukraine, there is no official registry. This is all voluntary, and not every clinic is performing their statistic and sharing their statistics. So real number is definitely more. When it is happening, because the average age for the first delivery in the world is increasing, and if the trend will continue, so we would need to have a decision as a treatment option for the patients in their over 40 and close to 50, in close 20 and 30 years. Most of our patients, which we see right now, are so-called poor responders. They are older, they have limited ovarian reserve, they have low IMH, and we are not changing our trends for the treatment as for the safety in IVF, but also we are creating different variants and strategies for the poor responders. However, they are ending up with the success rate between 3 to 10% in comparison to possible over 50 and 60% if they would use oocyte donation. However, for many of this treatment is just not affordable, and specifically now, as many patients from different countries needed to travel into different destinations also including Ukraine, in order to receive the treatment. We also created new strategies and techniques which are still experimental. However, they are very successful, and they are practicing successfully in Ukraine and also in our clinic as a spindle pronuclear transfer and mitochondrial donation. This option helps to those patients who are psychologically not ready to accept oocyte donation as getting materials just from the donor, who are looking for the option of using at least some of their materials. However, of course, oocyte donation, and we all know that is the most successful treatment nowadays. And even for those cycles, we also would need donor materials. Very few words about Ukraine. By territory, this is the biggest country in the territory of geographical Europe. We are more than 40 million people. We have about 60 IVF clinics. All are private. Oocyte donation and export of biological materials and also surrogacy are absolutely legal. This is written in the law and well regulated. And what is important for the oocyte donation, Ukrainians were recognized as the best looking nation in 2019. Also, our clinics and agencies were working as a source of the traveling donors for different destinations such as Cyprus, Czech Republic, Spain, USA and Canada, and some others, in order to send fresh donors to fly and donate in those countries. Also Italy and some other countries in Europe. However, this practice is also not possible right now. And in my personal experience and in the experience of the Ovogene Egg Bank team, we have practiced starting since 2008 of export biological materials from territory of Ukraine into different destinations, into different countries. And this is very well regulated. We know all the requirements and needs according this to happen, starting from the quality management, from the legal contract issues, and also the technical issues as supplying the transportation itself, custom documents, legal requirements, screening regimes for the donor in each location and each country. And in our practice, this is 38 clinics in all 5 continents already. In order to be compliant with the international regulations, we follow the regulation of the country. We have learned in details all the laws in every country, as I said, starting from the consents which needs to be signed by the donors, and the screening regimes, the testings which needs to be done for every donor. Either we have a need to have materials in the quarantine or not, etc. So Australia and Latin America, all European Union, and even countries inside the Union, we all know they are different country to country. Even sometimes the reading of the law in different clinics in one country is also different. However, knowing all the algorithms and learning the algorithms and having the decisions and the solutions, how to do this and experience, this helps this to happen really fast and easy. So the structure of the Ovogene Egg Bank is the following, that the main affiliated center and the clinic supplier is now located in Europe. This is IVMed clinic, which is located in Kyiv. However, according to the fact that we have strict processes and algorithms and requests for each clinic supplier, and also we have an IT created decision. This is special software where we control all the algorithms and processes as efficiency of the donor, as number of the stimulation cycles, as results of the survival rate and blastulation rate, as number of the children born for every location and every country. This is very easy to work with such a big volumes, with different countries, with different clinics, and follow different algorithms and make this all safe and reliable. If we are talking about donor advertisement, so we are starting from donor recruitment from the very beginning. We do that all by our team. We are not using third parties, any agents, anyone in that process. The main channel and very effective one is internet. We place the information about the possibility of being a donor on the website, social medias, forums. We do local recruitment, not only in the clinic, but also in the state hospitals by the doctors and nurses. We promote being a donor on radio and TV. And I should say that in comparison to the beginning of my career and my being involved into the oocyte donation back into 2008, it was, fact was that nobody knew what is to be an egg donor in Ukraine. And now this is absolutely opposite. Those women who are potential donors, they know about this possibility. They are just selecting a clinic for them. They are selecting a clinic they would trust, they would respect, they would feel safe there, and they would have relationships. So this is what is done by our team who is doing the recruitment. And very important is a recommendation. So bring your friend, is working really well. And this again says that donor trusts our facilities and our work. Motivation for the donor is of course financial, but I would say that this is for a small portion of the donor. And for most of them, we call this is altruistic and financial, because they really feel the responsibility, what is to be a donor, why they are doing this. They feel sharing something what they have with those who have a need to get this. Of course, the additional benefit is a fact that we perform complete screening, and this is for free. And in case any recommendation is needed, our doctors and nurses and medical coordinators take care about this. This is safe and reliable, which is also very important. And the most important probably is the fact that there is no waiting period. As we are working as an egg bank, and we need an oocytes every day, and we need big volumes. As soon as the donor is fully screened, we are starting her stimulation. These some results of our recruitment process just to understand the volume. During second part of 2020, 841 initial donor contacts have been made with our team. These are either phone interviews, phone calls, or online forms. During that time, we are asking very basic inclusion criteria as an age and the fact of having an own child. Those who have this, these are 841 donors. 319 were not accepting after personal, psychological, and social evaluation, or they refused on their own after learning all details of the program. 237 were not accepted due to medical reasons. Mainly this is the ovarian reserve and IMH level, or such factors as endometriosis or myomas. 4 were excluded after infections, disease screening, 9 after genetic evaluation and genetic consultation, and just 272 were accepted as our ovogene donors. So we are accepting about 3 out of 10 candidates who are entering our doors initially. And the algorithms is, as you could understand from the previous slide, as soon as we are getting the information about the candidate, we are providing complete information about the program to that person. We perform interview, we provide all the consents which she should learn and read and be agreed with. She signs the contract with the clinic and starts her screening. We train a donor how to make the injections and what is the influence of the medications. We plan her visit, perform egg retrieval, and pay her compensation. As soon as we got the biological materials, where they are placed on our online database. Our inclusion criteria are age of the donor between 18 to 29. There should be proven fertility of a donor, physically and mentally healthy, no hereditary habits, appropriate ovarian reserve, and this should be dependable, mature, and responsible person, which is also evaluated by the professional psychologist. And exclusion criteria are the facts that if there is no information about donor family history, for example, she was adopted, we would not include such a candidate into the donation. recommendations given by the psychologist or geneticist not to proceed with such candidate, positive drug test or infection disease, carriers of genetic mutation, or low survival rate of the oocytes in previous sowing cycles performed in 2 different locations. This is we analyzed already from our software, from all the data. In case we find such a donor, those oocytes are used only for internal use with the additional backup for the patient. Also, we analyzed 10 years of our database, very basic things, but they are also very important. So over 200 profiles analyzed for this year and over 2,000 and over 600 in 2010. So you can see that the parameters almost has not been changed. Just the average age of the donors right now in our database is 25.2 years old. It used to be a bit younger, 23.7. Average number of children now in our donor database is 1.2. And this is very common for Ukraine not to have a lot of children, 1 maximum 2. Average number before was 1.3. The age of the children we were not evaluated before. Now this is 4 years old. Average IMH is 5.3. Also 10 years ago, we were not evaluated IMH routinely for all donors. Our donors in database are mainly single or divorced, non-employed or working part-time, and their motivation we call as a compensated altruism. If we are going through the process of the screening, there is initial examination such as blood group, blood count, and the consultations of the doctors. After that, the hormonal screening and infection disease testing, which includes HIV, hepatitis B and C, syphilis, gonorrhea, chlamydia, cytomegalovirus, HTLV virus, etc. And the genetic counseling process, which includes interpretation of family and medical histories to assess the chance of disease occurrence or occurrence in the new family, 3-generation pedigree for the donors and its analysis, genetic screening recommendation, obligatory test, genetic tests, which we perform for all, are karyotyping, cystic fibrosis, SMA, Fragile X, phenylketonuria, and neurosensory deafness. Extended genetic testing for special cohort of donors, and we have in our database some donors which has already been extendedly tested, genetic test results interpretation, and providing the reports. So in our database, we have Caucasian, Asian, and African donor phenotypes. Of course, the biggest variety, these are Caucasian. Donors can be anonymous and non-anonymous, and this is dedicated specially, you can select this in the online database. We have transparent guarantees both for the oocyte survival and blastulation rate, and the booking and the access for the donor database is free. Also, we spoke about full donor screening and the specific embryological things as the unique genetically certified oocyte technique and partner clinic embryology lab supervision, which will be spoken more in details by Birol lately. And also, this is a complete decision for the clinics and the partners as a direct transportation of biological materials, but our safe and reliable partner, ArcRio. So how to start working with Ovogene Egg Bank? This is very easy. You shouldn't look for the solution by yourself and learn all the processes and the legislation. You should just contact us and ask us how to do this. We will be happy to guide and share our experience, which we already have. And we have that for most of the countries, as I said before. And in order to book the materials, you should register on the donor catalog and set up the free access for the donor profiles and photos. Choose and book your oocytes or donors and order the materials by courier service, including the date and time you would like them to be delivered for your clinic. Online database can be configured according to the request of your country and of your clinic. For example, for Australia, we have a specific database which is just for the patient from Australia, and only donors who has completed their initial testing with the counselor are included there. Or for example, for UK, we are placing the database without adult photos, as this is a request from the law of and the IGFA, and also donors are performing 2 PCR testing after the quarantine. So such materials are only available for this database. And these are more and more requests according to each country and those regulations. So the reservation of materials is absolutely free. You can select and reserve a donor at any time and at any place. Your reservation ensures that the oocytes are taken out from the gallery, meaning there is not that one materials can be booked by 2 different patients or clinics. You can choose the donor and the number of the oocytes you need for your treatment. And our team of the coordinators are always online to assist you with the donor selection and reservation. And if there was a mistake, for example, you are able to cancel that reservation. So we believe that the goals of reproduction are to achieve pregnancy as soon as possible, to maximize success rates for the first initial cycle of each patient, to reduce multiple pregnancies, to perform safety in IVF, and to help as many patients as possible by optimizing the cost and considering the low limits. And the last point, this is the point how Ovogene Egg Bank team sees the cooperation with their partners, with the clinics, also with the individual patients, by sending materials to the locations where you are and avoid traveling the patients, but let travel biological materials. By this, many clinics has already developed their long-lasting and very active and successful oocyte donation programs in their home facilities and in their clinics. So this is all I wanted to share with you during this evening, and I would like to invite Birol to continue and to share his presentation, and we will be happy to answer the question after both presentations are completed. Thank you so much.
Thank you. Hi, first of all, I'm glad to meet with you for the first Ovogene webinar in 2021. So I will try to talk about cryo donor egg banking efficiency for the Ovogene Egg Bank. Of course, first of all, I would like to talk how we are organizing our egg bank regarding embryology laboratory. So how we are doing a morphological assessment, how we are choosing a donor oocyte, which we are considering for egg banking. So we have, of course, a different criteria, like a general criteria, like cumulus oocyte complex morphology, zona pellucida morphology, periuterine space, first polar body appearance, oocyte shape. But also we have a difficult criteria than all other local and general egg banks. So we are using a meiotic spindle location for the morphological assessment. Also, we are using a time-lapse activity and artificial intelligence, and our unique product of genetic certified oocyte. So how we are using artificial intelligence in egg banking. Of course, artificial intelligence is a new trend of the IVF, but mostly you can see that they are using on the time-lapse activity for the embryo selection. But we try to develop also unique for Ovogene, how we can use artificial intelligence for oocyte morphology assessment. So as you see the image, artificial intelligence can measure different parameters on the donor oocytes. For example, thickness of the zona pellucida, like a space, periuterine space, size of polar body, diameter of the oocyte, and also cytoplasmic activity of the oocyte. So while we are working after the denudation with donor eggs, when we put under the active time-lapse modules, so there are different calculations for each parameters by the machine learning, and machine learning is just calculating a total diameter and size. So after this process, like all oocytes, donor oocytes, which selected by artificial intelligence, going directly for the meiotic spindle determination. We are using a special software and microscope objectives and polarization, so to determine exact location of meiotic spindle. Why we are doing that? Because abnormal meiotic spindle, as all publication shows, affect the chromosomal configuration and also increase aneuploidy rate for the oocyte. Why we are trying to supply for our customers best quality oocyte, but also best genetic potential of the oocytes. Of course, that is a question. While if we determine a meiotic spindle, how our final customer will get a benefit from this? Because still during fertilization with ICSI method, still there are possible damage to meiotic spindle. So how we solve this question? As you see on the image, we have exactly location for the meiotic spindle, which we draw with the graphic method. So it's coming directly from artificial intelligence. And when we send the oocyte our final customer, they will have same algorithm and they will find out totally under the microscope, their regular microscope, and they will guess where is the meiotic spindle. So they will avoid that part during the ICSI and fertilization. So this is the publication on 2010. So publish it, as you see, when spindle positive and negative, like if they determine the spindle, as you see, fertilization, day 3 embryo quality, blastocyst formation, post-towing and pre-towing always high when we determine the spindle. Also usable embryos rate and day 5 embryo transfer rate dramatically high post-towing and pre-towing when we find a visible spindle. So this is the algorithm how we are finding under artificial intelligence a donor oocyte selection. Doesn't matter how many donor eggs we have, when we put under active time-lapse camera, a time-lapse camera will collect with machine learning and software different parameters, even with the 20, 30 oocyte in a minutes. So that means like we are working under totally safety condition. So oocyte is not waiting like a 5-10 minutes under the microscope, so artificial intelligence can work. Finally, this is under the time-lapse incubator. So there is no any kind of morphological or lab damage during this assessment. So just in a minute, artificial intelligence calculate all parameters of the oocyte and finally give us exactly number. And if this number matching with our reference number, so this oocyte is suitable for the vitrification process. So as Ovogene Bank, we are only choosing a quality 1 oocyte which suggested directly by the artificial intelligence and which find out the meiotic spindle determination. So this is the image. While when we see really low quality oocyte, even artificial intelligence show us why this oocyte have low quality. So different cytoplasmic defects, double polar bodies, like a multipolar bodies, large perivitellin space, and cytoplasmic granulation. Of course, that in laboratory, there are many different embryologists. So many embryologists doing and joining this activity for vitrification and egg banking. So it doesn't matter how many embryologists has, but each microscope can collect this data and can give the final and main database this information. And as lab director, I am involving the process and to choose a final number of oocytes, which is going from each donor for vitrification process for egg banking. So if we talk about the genetically certified oocyte, which is unique product for the Ovogene Egg Bank, why we are doing such product? So this is the main thing. Many of the study shows still 20 of 30% of donor oocyte may have aneuploidy or different kind of chromosomal anomalies. So if we try to avoid this number, we believe that final customer will be more happy, because still if they get the 6 or 8 oocyte, still they have chance to avoid embryo NGS, and most of the final clinic or customer or patient, they are avoid NGS while they are doing egg donation cycle. They just do embryos and transfer on day 5. So still, if we are sending 6 oocyte, there is possibility that 2 of them may have aneuploidy if we calculate 20-30%. So we are just trying to avoid this risk. Of course, this risk, when we avoid, it gives big chance to patient to get 6 euploid or 8 euploid oocyte, which can give potentially up to 50% of a good quality of blastocyst. When we certify the oocyte, we are not only doing like a polar body biopsy and check NGS testing for the polar body. Also, we screen this donor with the carrier screening, and also we make target paternal risk analysis. So this donor and this oocyte will be totally safe as genetically, and we will certify this oocyte for our customers. So if we talk about our statistical outcomes, as you see, the oocyte survival rate for per clinic, while we have many different clinic final customer, we are working with them. Each of them, as you say, have different survival rate. So there are 19 clinics here, and as you see, some of them have very high survival rate, some of them have low survival rate, and some of them have average. Of course, that shows still that the final clinic and embryology laboratory, which is doing exactly this procedure, we should analyze their lab, their outcomes, their techniques, and we should include as Ovogene Egg Bank all process together with them. So while we are joining their process and we do lab audit exactly, we always achieve high outcomes, survival rate, fertilization rate, even we can affect the pregnancy rate. So I will show some statistic about the supplier of the Ovogene Egg Bank, because final supplier of the egg bank is very important point here, which can show us a direct information, because finally this supplier is preparing all oocytes for donor oocytes and vitrify, and in same lab, same embryologist, same techniques show us how is the main outcome for the survival rate, for the blastocyst rate, and fertilization rate. As you see from the main supplier during 2020, so that is a cryo rate of the blastocyst from the cryo donor eggs, almost 74% is really high. So from then, 10% are totally hatched, 8% are BB quality, 30% are BA quality, and 51% of the blastocysts have AA quality. So, and fertilization rate almost up to 90%. So that means that supplier clinics are using a stable techniques, good lab organization, and good towing process. So of course, we are trying the same condition for all of our final customers and to build with them high quality of collaboration. So this is the 3 different clinics. And first one, without transportation, is a supplier clinics, and 3 different clinics. They are survival rate, fertilization rate, cleavage, and utilization rate. As you see, without transportation, have a good survival rate outcome. Clinic 1, Clinic 2, Clinic 3 with the transportation also have really high survival rate. There are not significant difference between without transportation and transportation. Also fertilization rate, even some of the clinic which are using transportation have higher fertilization rate than without transportation. If we check about the cleavage rate, almost for all 3 clinics and without transportation supplier clinic have similar cleavage rate. So that shows like when we build good collaboration and good lab audit, if we use a strictly towing protocols as same protocol with each clinic and final supplier, we will never have questions about survival rate and outcomes. So if we check the supplier and 2 different clinics, so supplier, if we are talking about 240 cases, clinic A 65 cases and clinic B 45 cases, which use cryo-donor eggs from Ovogene. So average of the number of cryo-donor eggs, supplier 8, clinic A 12, and clinic B 8.5. So for each cases, they use this average of the all site. If we check the survival rates, so they are all supply up to 90% of the survival rate. Fertilization rate also similar. Blastulation rate, if we check, supplier clinic have bigger blastulation rate than others, and pregnancy outcome almost high for each 3 clinic, because if we check the average age of patient which use for the embryo transfer, so almost up to 40 years old. If we check on the graphics, so while we see when survival rate is high and fertilization rate is high, also blastulation rate is really high for 3 of the clinic, which has more cycle than other clinics. So, and if we check the pregnancy rate, supplier clinic A and clinic B has really high pregnancy rate, even such high age group. This is very important data, I think will be interesting for all of you. So if we check NGS outcome from the cryo egg banking, which use cryo egg donation program, so supplier clinic has 65.70% of euploid embryos, which they receive from the cryo donor eggs. So almost very low mosaic rate and non-diagnostic embryo rate. So if we check another clinic, which we sent a transport eggs, so also they have 49.48% of euploid embryo rate, which is quite well. And another clinic also, which received by the transport cryo donor egg, almost have 54.81% euploid embryo rate, which is really nice. So what is unique for Ovogene Egg Bank? And finally, so we have unique vitrification and warming protocol, which we developed by ourselves with according our experience, many outcomes, donor efficiency, and while we are using artificial intelligence and other data. So safety transport by professional medical couriers. This is one of our proud parts, which we trust so much. Active lab support and audit. So we are open to communicate anytime with all embryology laboratory, all embryologists and all lab directors. Donor selection regarding efficiency. This is really important. We have special software which we can get all parameters. And finally, in supplier clinic, we have many IVF cases. So for each donor, we have idea about their blastulation rate, fertilization rate, pregnancy outcome, and also euploid blastocyst outcome, which is very important for the donor efficiency. And of course, we are using technology based of morphological assessment, as I told, like artificial intelligence, meiotic spindle determination, and from the, I guess from the end of February, we will be able also to supply even our final clinics, like a live video of the vitrification process, live image from the time lapse and artificial intelligence. So they will be able to totally trust what they are towing. They will be sure about the quality of the oocyte, what they are selecting from the ovogene egg bank. Also, our unique service, genetic certified oocyte, we will show during 2021 all article and publication regarding genetic certified oocyte and artificial intelligence outcome. So thank you for your time. And if you have